Hello friends, welcome back to my channel Biofreak. Let's talk about polygenic inheritance. In polygenic inheritance, we see that many genes control one single character, or I can call it one single trait. So in polygenic inheritance, there are three important things which are to be remembered. So first thing is that there is no dominant allele. Okay, so there is no law of dominance which is followed, neither there is any recessive allele. So basically I can see that polygenic inheritance is violating your law of dominance and basically your normal Mendelian rules. Second thing is there is also no epistasis which is considered here. Epistasis is basically when two genes are interacting, one gene can inhibit the other genes. So basically that was your epistasis. And the third most important thing is that there is something called as additive phenotype. So what do I mean by additive phenotype? Let's take the dry weight of a particular plant. Okay, I'm considering dry weight of a plant. And this plant has two alleles, capital A and small a. And these two are contributing to the dry weight of the plant. So capital A allele is responsible for adding 2 grams to the weight, and small a allele is responsible for adding 1 gram to the weight. Just an hypothetical example, okay, to understand your additive phenotype better. So if you consider when plant gets capital A and capital A allele together, the addition of these will lead to 4 grams of weight. If you consider capital A and small a together, it will be 3 grams because it's 2 plus 1, 3 grams. And small a allele, when these two alleles come together, although there is no recessive allele, you're getting the addition which is 2 grams. So here you can see that there is no dominance or recessiveness which is seen, but there is addition of each allele which is contributing to the same trait, right? So now let's look at two more important terms. There is discontinuous variation and there is continuous variation. When we say discontinuous variation, you can talk about this tall and dwarf characters, right? Or these two traits were considered by Mendel, which were actually wrong. Why? Because there can be some intermediate height as well. It's not extreme tall or extreme dwarf. In between also you can get multiple different heights, right, which exist. So this was actually the part of your continuous variation. So your height, foot length or hand span, all these are in continuous variation. Even their graph, if you consider, is a bell-shaped curve. Your height, weight, heart rate, finger length, leaf length, whatever there is in terms of length and weight will be in the continuous variation. So you cannot get, like, there are no two extremes, either you're 60 kgs or you're 20 kgs. That's not these in between also different weights exist, right? So that is continuous variation. What is discontinuous variation is where you get this type of bar graphs. Example here is eye color, gender or blood group. If you consider blood group, there are only 4 different A, B, AB and O, right? So this is discontinuous. You won't get this type of graph here. It is bar graph. And ability to roll the tongue. Either you can roll the tongue or you cannot. So these are the two different things, and there is no continuation in between, right? So that is your discontinuous variation. So I can say that continuous variation is related to your polygenic inheritance, right? In polygenic inheritance, we can see that, for example, your height is controlled by multiple genes, okay? And under continuous variation, you can see that you will get this type of graph. So let's look at two different examples. The first example here is of wheat kernel color. The point here is that when they made this particular cross, between the dark red and white parent, they got medium red, okay? And there was a variation. They got the ratios as 1:4, 6, 4, 1. Let's learn two important formulas before going in deeper, okay, in detail. So let's talk about first formula, which is important. To find out how many different genotypes you can form, you need to follow this particular formula, that is 3 raised to n, where your n is number of different alleles or different loci, okay? That will give you the number of genotypes, okay? And the second thing here is to find out your extreme phenotype what you can do is that you can use this particular formula, that is 1 by 4 raised to n, where n is again the loci number. Now let's try applying these formulas in wheat kernel color example. So here, can you see how many, you can see that there are approximately, there are 9 different genotypes which are there. Now let's utilize this particular formula here, 3 raised to n, where I can say that how many loci are there. I can see there is R and small r. It's all about 2 different alleles here. So I can say that it's 3 raised to 2. So I should get 9 different genotypes, which is true. I can see 9 different genotypes. 1, 2, 3, 4, 5, 6, 7, 8, 9. Now, they got this particular ratio. They got just 1 for dark red. Then they got 4 for medium dark red. Then they got 6 offspring for medium red. Then they got light red 4 offsprings and 1 which was white offspring. So total, how many offsprings were produced? 16 produced, right? But extreme phenotype, to find extreme phenotype, I can use this particular formula, 4 raised to n. N is again 2, so 4 into 4, that will give you 1 out of 16 phenotype will be your extreme phenotype. So this is how you can use another important formula there. So I hope you understood what was related to this particular kernel color. We have just 2 different alleles. It can be also given like this. So there are 2 different alleles crossed with small a, small b, small b. Now you can see that there are just 2 different loci, right, which are involved here. So that's why I am adding n as 2, and you will get number of genotypes. And this particular ratio is again important. And here, when you plot the graph, you will get a bell-shaped curve, which will be 1 ratio 4 ratio 6 4 ratio 1. So this is how there is kernel color, wheat kernel color example which is given. The next is wheat color. In wheat color, there are basically capital A, capital A, capital B, capital B, capital C crossed with small a, small b, small b, small c, small c. So there are 3 different loci. Directly I can use this particular formula and I can add 3 raised to 3. So I can expect 27 different genotypes. How about extreme phenotype? It will be 1 by 4 raised to n. N will be again 3, right? So it will be 1 by 64. So total boxes, if you count, they will be 64, and 1 will be extreme phenotype, which is white given here. Okay, and at the same time here, another extreme that you can get is deep red. So there is a variation. There is no distinct thing here. There is a continuous variation in this particular table. You can see, okay, this is the normal Punnett square method. If you want to solve it, you can solve it by yourself, but it is just waste of time. You can just look at this particular table, learn the ratio. Ratio is important. They are getting this ratios, okay, like this. So they got deep red was 1. Then they got 6 that were dark red offsprings. Then they got 15 red offsprings. Then they got in between medium red. Okay, so when you draw this particular graph, it will be something like this. So 1 was your deep red, then came your 6 which was dark red, then 15 offsprings were red, medium red was 20 offsprings, then again they got 15 offsprings, then they got 6 offsprings which were very light red, and then finally they got just 1 white. So they are going from dark to very light color, and there is a continuous variation. This is another example for your polygenic inheritance, how the color is imparted in this particular wheat seed. Okay, so this is an example of wheat color. Okay, this is for wheat color. The previous one was for the wheat kernel. Okay, this outer coating that you can see now, that is wheat kernel. And wheat actual color is given here, that is your deep red till white. So there is a variation. The ratio is 1 ratio 6 ratio 15 ratio 20 ratio 15 6 1, like that. And here it was this particular ratio. So I hope you understood this particular lecture and you found it interesting. See you in the next lecture.